Hi, my name is Antonio Rua, and thank you for viewing my presentation. I did my research looking at the gp73 function in the bacteriophage MosMoris. So, a bit of an abstract about my project. The groundwork of this was done using synthetic biology. So synthetic biology is this new and emerging field of biology that kind of takes a bit from a whole bunch of fields: biology, engineering, systems biology, biotech, bioinformatics, chemistry, math. All of these kind of contribute a little bit to what synthetic biology is, which I'll go into a little bit more later on. So, like I said, it was most of the groundwork was done using this synthetic biology in order to research this gene gp73, which, while it's well known across a bunch of different bacteriophage species, the function of it as of so far is completely unknown. I then took the gene encoding for the gp73 protein and I redesigned it in order to make it not functional and not work in the virus. Through different analytical, mostly bioinformatical at this point, techniques, I found that the gp73 functions most likely in the replication of the virus, but there's still more tests that need to be done. And most of this are using more wet lab techniques to assess the function, which I'll go into a bit more detail later on. So on the screen right now, you see the typical life cycle. Viruses aren't necessarily alive, but if there's anything that's close to it to call it that, it's, I guess, the life cycle of a bacteriophage. And so in the first step, the bacteriophage attaches to the bacterial host cell, and then it inserts its DNA, and then it kind of hijacks the cellular machinery of the bacteria in order to make more of it and assemble more of it. And once enough are grown up, it breaks out of the cell, killing it. This research kind of had two goals in mind. The first was to create a non-functional gp73 strain of the virus. That way we could assess the function in its absence. The second was to assess the function. Most of the groundwork was developing this non-functional gp73 gene and growing it up, and we'll talk more about what still needs to be done later. So a little bit of background on bacteriophages. They're very, very small viruses. They only infect bacteria, so they're gonna have to be smaller than bacteria. And they have been shown to effectively kill drug-resistant bacteria. And bacteriophages are being used more and more in bacteria-targeted medicine. It's a new emerging field of research being done. And bacteriophages are made essentially out of three main pieces: the capsid, or the head, that contains the genetic code of that virus, the collar that attaches the head to the tail, and the tail, which is essentially this hollow tube that the DNA goes through when it's injected into the cell. So using synthetic biology, you can utilize and rewrite and reprogram DNA. So you can cut out junk DNA that doesn't really matter. You can optimize things within the cell, or, as I've done, you can research functions by creating just new versions of that DNA. As I mentioned, gp73, while well conserved, has a completely unknown function. The MosMoris, the bacteriophage that I use, is relatively recent. It was found by Dustin McHugh and Milbridge Maid in 2012, isolated from the bacterium Mycobacterium smegmatis, and was only really fully sequenced on January 25th, 2013. And when I say non-functional, some of you might wonder quite what that means. So I substituted in a premature stop codon, which stops the function of the protein. And as you can see on the screen, to the right, you have this extra stop that's just kind of in the middle of that sequence, and it stops the entire DNA sequence from being read, which when you have something that's a half or a third of the size, it's not really going to be able to function. So a little bit of how I did the research. First, I did a BLAST-P search with the MosMoris gp73 sequence, essentially trying to compare it against known in the database sequences that are similar to it. And I picked out a couple of phages, Marvin, Vasuzinga, Tesla, and Typha. And I took those sequences from those phages and I put it into this program called Cluster Omega, which you can see on the right. It essentially just lines them up so that you can easily look down the columns and compare them. I then chose strings of amino acids that were conserved or the same through all the different phages, and I took the long chains of those conserved amino acids and started to substitute them out, hoping that they would be important in some sort of binding pocket or functional domain, as there is probably a reason why 6 or 7 amino acids in a row are perfectly kept throughout all the phage species. And those conserved amino acids are the ones with the asterisk right there. You can see them being the same down the line. I then took it to Johns Hopkins gene design software, which essentially just lets you build these things called building blocks, where you can break the DNA sequence up into these little bits, and then based on where they overlap, you can stitch them together to make one large custom bit of DNA. Then using EcoR1, essentially like DNA scissors, it's a restriction enzyme. I cut open the plasmid vector, JO4450, and I just inserted that building block that I designed. And then I inserted it into bacteria, and this is a method called Gibson assembly. I then took this plasmid, inserted it into some E. coli, and then I plated them on plates with ampicillin, antibiotic, and I grew them up. On top of this plasmid, as you can see in the top right, there was a gene for antibiotic resistance. So that meant that any bacterial colony, any bacterial cell that picked up this plasmid with the new gene inside of it would also be able to survive the ampicillin on the test plate. So I would know that any bacteria there would have this new plasmid in it. So a little bit about the results from this. When we compared the amino acid sequence of the MosMoris gp73 against a database of known proteins, we actually found that almost all of the proteins that we matched it up against functioned in some way to replicating the virus, or just being some form of effector protein. And so this is what we kind of are using as of right now to say that it most likely functions in the form of replication. every known, as you can see with this first match, which is 99.16 probability, a essentially 0 E-value. Most likely, this protein will work either affecting and causing a cascade for replication or organizing that replication machinery. And then we have the bacterial transformation and replication results. As I mentioned, we had antibiotics on those plates. And as you can see from the test plate that we would then select colonies from, it's not a bacterial lawn. There are clear identified colonies that survived that ampicillin, and there was a lot of them, which was good. It means that a lot of bacteria picked up that plasmid. You can see some of these red or pinkish colonies, and that means that those bacterial cells picked up the plasmid, but the plasmid didn't necessarily pick up that gp73 gene back when it was being done in the Gibson assembly. So these colonies were just avoided when we chose colonies for storage. On the negative control, these were colonies that didn't get the gene for antibiotic resistance. When we retested these colonies with more direct antibiotics, they died out. So we just kind of chalk this one up to experimental error, especially considering that we know that the antibiotics worked, because this bottom right plate had no antibiotics, and while it looks like nothing grew, it's actually just a completely clear film of straight bacterial growth. In this bottom left plate here, you have the positive control. You have colonies that we knew had the plasmid. They already had the antibiotic resistance with them, and the growth looked very similar to our test plate in the top left. As I said, it was a little unexpected how much growth there was in the negative control, but it was significantly less than either the positive control or the test plate. Then, unfortunately, the end of the semester came, and so it came time for storage. And so we took some colonies that, as I mentioned, were the white colonies on the test plate, not the pink ones. And we stored them up with glycerol and some bacteria, and we put them in the minus 80 degrees Celsius freezer to kind of cryo freeze them for long-term storage. Going forward, they just need to be thawed out of the minus 80 freezer and use that bacteria to stitch together the non-functional gp73 variant with the rest of the normal MosMoris genome. And then from there, you can grow up a lot more of those bacteriophages and perform tests on the function of gp73 specifically. We can electroporate cells to help them kind of get into it, and then perform what's called a titer, where you just serially dilute. You take 0.1 milliliter of the stock of virus, and then continually just take 0.1 milliliter and keep making times 10 dilutions. And you can use this to test for virality, because if the normal MosMoris at 10 to the minus 5 looks like this, but the non-functional gp73 at 10 to the minus 5 looks more like this, and there's way less of those viral plaques on it, then we can see that clearly gp73 is needed in order for this virus to be truly viral. And that's all I have. Thank you for coming to watch my talk.